I am going to start talking now. Aloha. Welcome to the last day of PSB. I'm Russ Altman, one of the co-organizers. We have been very pleased with how the meeting has gone, and we thank you all for your participation over the last couple of days. Yesterday was a particular challenge for all of us because during the poster session, the US Capitol was attacked. And so I just wanted to acknowledge that terrible event and thank everybody for their patience as we tried to muddle through during what was the most distracting of times. As you know, the PSB doing it virtually was not our first choice this year. We do expect it to be on the Big Island again next year for number 27. Please consider session and workshop proposals. This meeting is good to the extent that people are thoughtful and send in the best proposals for emerging areas in computational biology and bioinformatics and clinical informatics so that we can have this meeting, which is always dealing with cutting edge issues. The instructions for websites, for workshops and for sessions are on the website. Under these very difficult circumstances of a remote PSB, I want to especially thank the staff, Tiffany, Cindy, and Ryan. They had to figure out how to do this. After doing this meeting for 25 years, we have a way to do it, and it's almost a reflex. And all of those reflexes went out the window this year, where we figured out poster sessions and workshops and how to do it. So Tiffany, especially, and as well as Cindy and Ryan, just really stepped up, and we're very grateful for that. And we hope to have a non-virtual meeting next year. So for today, there's one session coming right up in a moment. And then I just want to highlight that there are 5 workshops which will run in parallel, starting at around 10 a.m. Pacific. I think it's, Pacific. And then you'll have to do the math for your, and these are on great topics. Very quickly, bioinformatics of corals, translational bioinformatics, making tools that people will use, improving patient outcomes through collaboration, and establishing the reliability of algorithms. So really good topics. And all of the URLs are in the program that has the URLs in them. So thanks again to everybody. I hope you're all safe. I hope 2021 is much better for you, or at least as good as 2020, if you had a good year. And now I want to throw it to Sam, one of the organizers.
All right, thank you very much. So I'll give a quick introduction here. We're gonna, our session's on biocomputing and AI for infectious disease modeling and therapeutics. And go to the next slide. The co-chairs are myself, Waylon Alterowitz, Gail Castle, Lixin Zhang, and Keith Dunker. One of the things I just wanted to quickly note is just that to dedicate this to Marco Ramoni, who is a big proponent of PSB and passed away in 2010. This is actually a picture from 2010. I was writing his initials on the beach at PSB. And it was during that time actually that I met Waylon. And it may be the first marriage that resulted from PSB, I don't know. But this past year we had Avi, and his middle name Marco also is to commemorate Marco Ramoni. So I just want to thank him for all of efforts and support and for PSB and beyond. So with that, I just wanted to give a little bit of background. When we submitted the proposal, it was around, I think, January, and COVID was just really starting then. But we're thinking about infectious diseases in general, and COVID was one of those. So HIV, Zika, Ebola, other bacteria. And one of the ideas at that point was thinking about resistance as being a challenge. And it has been in different bacterial and viral strains. We're starting to see some of that also now, potentially with COVID, with the South African strain and so forth. So there are a number of different computational approaches and different ways to think about modeling, including disorder protein modeling. We have a couple of papers on that, and a number of different approaches ranging from protein-protein interactions to new methods and tools and pipelines and their associated algorithm development. So in the last kind of, I guess, 5 minutes or so, I'll kind of very briefly just highlight the different papers and point you to a few of them that are also in the proceedings in the poster session. So the first one, which we just started to hear a little bit of the beginning of, was the TreeFix TP, which is Sam's going to be talking about, which is a new method for reconstruction transmission phylogenies, and applies that in HCV, and is interesting work and has some of the code available for people to look at. We will then hear a talk that focuses on social media and COVID-19, characterizing physician perspectives and looking at the social media, comparing different perspectives on COVID-19 using that medium and methods that can do that by Catherine. And then finally, in the talk section, we'll be looking at protein-protein interactions, and that will also look at SARS-CoV-2 as well, and that will look at a number of the different interactions between different viruses and their host, and that'll be presented by Meghana. We also, as I mentioned, have a few posters looking at different aspects, so the biomedical literature and COVID-19, plus it also looks at some neglected tropical diseases. And that'll be exploring the biomedical literature using different algorithms there. And then there's another paper looking at disordered regions, or there are a couple of papers in this section about looking at intrinsically disordered regions. And one of them will be, the poster was presented actually yesterday by Anish, and looking at drug discovery by focusing on these regions and thinking ahead and analyzing, in that case, nearly 300,000 different compounds. The second poster on intrinsically disordered proteins, which I believe this topic had a full session on it in the past PSB, but in this one, the focus is on looking at SARS-2, and it found, and it characterizes this organism as having exceptionally hard outer shell, and looks at the implications of that for the immune system for developing different vaccines and therapeutics, given that analysis. So with that, I'd like to turn it over to our first speaker, who will be Sam, to talk about TreeFix TP. Once he's done, we will have a short, approximately 5-minute period for questions, and those can be posted, and we'll go from there. All right, so I'll pass it over to Sam then.
Hi, my name is Samuel Slajewski, and today I'm going to be presenting our work on TreeFix TP, which is software that we've developed for reconstruction and error correction of infectious disease phylogenies using transmission network information. So the motivating problem here is the reconstruction of infectious disease networks. That is, given a set of individuals, determining the edges or the transmission events that caused the disease to be spread between them. This is valuable from a public health standpoint because understanding how and where the disease is spread allows you to develop strategies to combat that. For example, knowing that the coronavirus was airborne and oftentimes spread indoors led to mask wearing and restrictions on indoor dining. So there are a couple strategies to approach the transmission network inference problem. You can do things in person, such as interviewing and contact tracing, and you can also take a computational and molecular approach where you sequence strains of the virus from individuals and construct phylogeny, or an evolutionary history of those viral strains. Then, because you know the labels or the hosts that those viral strains came from, you can label internal nodes of that phylogeny and infer a network based on state changes. For example, in the cartoon here, a transmission between host B and host C would be indicated by an edge in the phylogeny with a blue node upstream or closer to the root, and a red node downstream or closer to the leaves. Some drawbacks of current methods for phylogeny-based network information fall into one of two classes. Methods which try to first estimate a phylogeny and then estimate a transmission network on top of it, are subject to errors in the original phylogeny construction due to the large space of possible phylogenies on sequences. If there's insufficient sequence information to construct an accurate phylogeny, long or short branches can lead to incorrect network inference. Methods which try to circumvent this by inferring both a phylogeny and a transmission network together often use sampling approaches like Markov chain Monte Carlo, which fail to scale to larger outbreaks or longer sequences. Finally, almost all such approaches only use a single sequence per host, whereas with recent decreases in cost of sequencing, we can now sequence multiple strains from an individual, for example, when they come back for a retesting. And because we know that viruses actually continue to evolve within individuals and there is an evolutionary development within individuals, sampling multiple strains from an individual host gives additional information. So we developed TreeFix-TP, which is a computational method for error correction of viral sequence phylogenies. We have accurate reconstruction of phylogenies and increased accuracy of outbreak and transmission inference while maintaining a scalable analysis for larger numbers of viruses, larger numbers of hosts, and longer sequences. The approach we use is we take advantage of high throughput sequencing to sequence multiple strains from each individual and then error correct a maximum likelihood phylogeny. TreeFix is available at the link here, which I'll also put in chat at the end of the talk. So the problem statement here is given multiple viral sequences from infected hosts and a starting phylogeny estimated on those hosts using something like maximum likelihood, we want to create an error-corrected evolutionary history where leaves are labeled with infected hosts. This can then be used to infer a transmission network by that state change method. So the idea for TreeFix-TP is based off of the idea introduced in TreeFix, which is when you're searching among this tree space, the primary method by which you evaluate trees is their likelihood with respect to the sequences. But there are also other potential ways to evaluate a tree. And it's possible that you have another tree, T star here, that is within some small delta in terms of likelihood, but is considerably better by another metric. In TreeFix, this was the reconciliation cost between a gene tree and a species tree. In TreeFix-TP, we determined the best tree by looking at all high likelihood trees and selecting the one with the lowest transmission cost. Transmission cost is calculated by labeling leaves with associated hosts and then using Fitch's algorithm for the small parsimony problem to calculate the minimum number of transmissions that phylogeny would require. For example, in the figure below, you have 2 orange hosts and 2 blue hosts. With the topology on the left, only a single transmission is required to explain the host pattern, whereas on the right, 2 discrete transmissions would be required to explain that phylogeny. Fitch's algorithm is nice because it's computationally scalable, so it runs in O of nk time with n leaves and k hosts. And this transmission score metric is biologically meaningful because by minimizing the number of state changes along a phylogeny, we're directly minimizing the number of transmissions that would be required to explain the phylogeny. So by the parsimony principle, this lower cost tree is more likely to be accurate. We evaluated TreeFix-TP using 2 sets of data. The first was data from a set of HCV outbreaks. We had 33 populations for which we knew the true host or the true source of those outbreaks for 10 of them. When we reconstructed phylogenies for these outbreaks using RACSML, which is a state-of-the-art method for maximum likelihood phylogeny reconstruction, and then error corrected those trees using TreeFix-TP, what we found was that in all 10 outbreaks, the tree returned by TreeFix-TP actually had a lower transmission cost than the RACSML trees, so they required fewer transmissions to explain them. Then, when we used the second half of Fitch's algorithm, which actually labels internal nodes of those trees, it's not a super sophisticated way to infer transmission networks for larger phylogenies, but for smaller trees, this is a good way to label internal nodes, and we identified the host at the root of the tree, or the source of the outbreak. Using the RACSML tree to identify the source of the outbreak, we only found the correct root in 6 or 7 of the outbreaks, depending on the routing method used. Whereas using the error-corrected tree from TreeFix-TP, we're able to identify the correct source of the outbreak in 8 cases, regardless of routing. We then tried to apply TreeFix-TP to a large set of simulated data to assess the impact of different parameter sets and different evolutionary rates on the power of error correction. So these data sets were simulated with FAVITS, which is a framework for simulating viral transmission and viral evolution developed by Nemo Moshiri. We used standard parameters for transmission network simulation, the Barabasi-Albert model for contact network, the SEIR and SIR compartmental models to actually simulate transmissions. And then evolution was simulated using a coalescent model with the GTR plus gamma sequence evolution model, where parameters for these were actually estimated from our prior HCV data to make sure the viral sequences were realistic. Within this simulation framework, we varied the length of the sequences, the number of viruses that were sampled from each host, and the scale factor, which is a proxy for the height of the tree or the rate of evolution. And we evaluated all of these different parameters within this analysis pipeline, where we simulated a transmission network and a phylogeny. We reconstructed a maximum likelihood of phylogeny using RACS-ML, which is again a state-of-the-art method. And then we error corrected those phylogenies using TreeFix TP. We then took the RACS-ML and error corrected phylogeny and compared them with the true tree using the Robinson folds distance, which is a standard metric for distances between trees. What we found was that on average, the TreeFix TP tree had about 7% lower transmission cost and was also 8 to 10% closer to the true tree. So the average RF distance per leaf from the true tree for RACS-ML reconstructed trees was 0.15, whereas the error corrected trees were an average of 0.13 RF distance from the true tree. When we looked at the impact of varying a bunch of different parameters, what we found overall was that error correction performs better or was more important in the presence of less sequence data. So for shorter sequences and for fewer numbers of viruses per host, there was a larger difference there was a larger increase in accuracy as a result of error correction. Whereas for longer sequences and more viruses used, the RACS-ML tree, the tree that was reconstructed using purely sequence information, tended to be about as accurate as the error corrected tree. We also found that the rate of evolution didn't have a huge impact on the ability of TreeFix TP to perform error correction, but we found that across almost all parameter settings except one scale factor, TreeFix TP was significantly more accurate than the RACS-ML tree, and for the single parameter setting, both trees were kind of statistically equivalent. Future work in this direction would be the application of TreeFix TP to the coronavirus phylogeny. We're looking for data sets where individuals have had their viruses sequenced multiple times in multiple tests so that we can apply this framework. We also are looking for strategies to reduce the overall error of first phylogenetic inference and second network inference by further applying parsimony principles. And we've actually developed the software TNet, which uses a parsimony-based principle to label internal nodes and infer transmission network phylogeny. Finally, there are applications for this type of work in domains beyond virus transmission, such as tumor evolution and tumor migration. In conclusion, I'd like to thank my collaborators on this work, Cheng-Chen, Makul, and Jan. And I'd also like to thank Alex Zelikovsky, Pavel Skums, and Yuri Kudrukov, who provided us with our HCV data from Georgia State. Finally, thank you to the funding sources and to the rest of the Bonsall Lab, and I'd be happy to take any questions in the Q&A. Thanks.
All right, so it looks like now we're in the Q&A period, and would like to see, we've got a few questions already. So we've got a question here around, have you applied this to B.1.1.7, and did it agree with the published origin account?
Yeah, so we haven't actually applied this method to any of the COVID strains. As I mentioned, one of the key things that we take advantage of is multiple sequences from each host. That's important for calculating this transmission cost. And we haven't really been able to find any data sets that allow us to do that. I'm sure that they're out there somewhere. I just haven't applied it yet.
Yeah, okay. Yeah, so that sounds like some interesting future work there. We have another comment/question. Recent work has shown that different variant calling methods lead to different transmission inferences and phylogenies. Did you test different SNP calling algorithms, and did they lead to different inferences?
So we didn't do SNP calling for this. We just used kind of the raw sequences and computed a sequence alignment from the raw sequences and then looked at state changes along that.
Okay. Let's see. We have another question there.
Sorry, my headphones came off for a second. Could you repeat
I
that?
have another question. Will COVID sequences from GIS-AID be sufficient to run TreeFix TP? They've assembled nucleotide sequences and subject metadata.
Yeah, I'm sure if there's, so kind of the only thing that's required is, we tested on sequences of up to length 1,000, so it'd probably be best to run for like a single gene family rather than the entire genome. But as long as the metadata is there, kind of linking the sequence to the host, it should be able to run fine.
Okay. Well, if any other questions, let us know. Otherwise, we do have another question. In the context of coronavirus, there may be convergent evolution under pressure of the vaccine. How would this method work in that case?
So by convergent evolution, that you're saying like the strains look similar, they kind of pick up the same qualities even though they're not evolutionary related. And I think this would actually be, I think this would be especially helpful in that case, because with convergent evolution, the actual sequence information could be confusing. You could have two sequences that aren't evolutionary related, but that look like it when you're just looking at the nucleotide sequences, and so you put them close together in the tree. But if you actually know the host that they come from, then you can, you'd be able to differentiate more clearly that they are not close together in the tree.
Yep, right, perfect. All right, well, that's a lot of potential there, and looking forward to seeing how that goes. So let's go then. We're ready then for our next talk, and that is going to be by Catherine, characterization of anonymous physician perspectives on COVID-19 using social media data. So I'll pass it to Catherine.
Hello, I'm Katie Sullivan, a postdoc at the University of Colorado. I wanted to first and foremost thank you for allowing us to share our work here and take part in this really great conference virtually and safely. Today, I'll be talking about some work we did using social media data, specifically Twitter, to characterize anonymous physician perspectives about COVID-19. So understanding physicians' perspectives about the COVID-19 response helps us to identify opportunities for immediate improved response to the COVID-19 pandemic, and may even help inform an improved response to possible future pandemics. Physicians have unique frontline experience that lends insight into the impact that COVID-19 has had on frontline healthcare workers, patients, and even the public as a whole. These insights might in turn lead to improvements in health outcomes, inform better health policies, and can affect health systems' safety culture, which directly and indirectly impacts patient health outcomes. In our work here, we wish to gain some insight into these physician perspectives surrounding the COVID-19 pandemic by using social media data, specifically Twitter, which with approximately 330 million active users per month has become a valuable resource for evaluating current public discourse and sentiments. So many physicians are actually hesitant to publicly discuss topics such as lack of sufficient PPE, testing equipment, or other issues surrounding their COVID-19 experiences for fear of being reprimanded or even let go. And this makes collecting honest representations of physician perspectives on social media difficult. Fortunately, in light of those concerns, a specific user account was somewhat uniquely set up where administrators collected direct messages from physicians and posted them to this page on behalf of those physicians, giving them some anonymity and a platform to express their COVID-19 perspectives. We extracted tweets from this page that were posted between March 16th and July 17th. All tweets were in English. And to prepare the tweets for analysis, punctuation, special characters, URLs, and stop words were removed, and words were lemmatized, and we ended up with 875 tweets. We also wanted to see how these physician tweets compared to general COVID-19 chatter on Twitter. So we used a publicly available large-scale Twitter data set composed of about 513 million tweets containing COVID-19 related discourse. And this data is actually maintained by researchers at Georgia State, including Juan Banda, one of the wonderful collaborators on this project. We then counted frequencies of the most common bigrams and trigrams in the physician tweets and compared them to the most frequent bigrams and trigrams in the larger COVID-19 chatter Twitter data. With the help of an experienced hospitalist physician, we also identified 4 specific topics that were recognized to be important within physician and public discourse. These phrases were personal protective equipment, unemployment, telemedicine, and racial injustice. We measured the frequency of these topics and the sentiments of tweets containing phrases related to these topics. So in addition to just counting frequencies of topics in the tweets, we also wanted to evaluate sentiments expressed in the physician tweets, and also garner some insights into whether certain sentiment analysis tools could accurately assess sentiments of these unique tweets. We chose to use Nrc and Vader, and both are unique in their own ways. So unlike other sentiment analysis tools, Nrc measures 8 emotions, including anger, anticipation, disgust, fear, joy, sadness, surprise, and trust. It also measures 2 sentiments, negative and positive. Vader, on the other hand, is a sentiment analysis tool specifically designed for analyzing social media data, and using a lexicon and rule-based approach provides a compound score between -1 and +1, and provides a matching categorical sentiment of negative, neutral, or positive for each tweet. And since Vader is catered more towards social media data, we used results from this method to compare to the larger COVID-19 discourse data and to calculate accuracy measures against manually classified tweets. So on the left are the most frequent bigrams and trigrams for the physician tweets, and on the right here are the larger COVID-19 data. Phrases from the physician tweets like healthcare, COVID patients, need PPE, and surgical masks are more specific to healthcare and being on the front line of the pandemic. While more general discourse in green on the right are more general, but also contain more political phrases like White House, Trump administration, and Dr. Fauci. So when we look for the 4 specific topics of interest, PPE, unemployment, telemedicine, and racial injustice, there were a higher percent of tweets that mentioned PPE, with the least mention surrounding racial injustice within the physician tweets. And then although all of the percentages are quite small, this kind of flips in the larger, more general COVID-19 data, where the highest percent was seen for racial injustice and the lowest percent was seen for PPE. So for the Nrc sentiment analysis method, we can see that overall it found more positive words than negative, and trust was the most frequent emotion identified in the physician tweets, with anticipation and fear coming in second and third. And upon manually reading the physician tweets, I would say that trust was not necessarily the emotion that best describes this set of tweets. I'll touch on this again in the discussion, but there seems to be room for improvement here for the sentiment analysis tool with regard to this unique type of social media data. Now moving on to the Vader sentiment analyses. These figures show negative, positive, and neutral sentiments over time for the physician tweets on the left, and for the larger COVID-19 data set on the right. In the larger COVID-19 data set, there were slightly more positive tweets over time compared to negative tweets, while there is less distinction among the physician tweets. Of course, there are far fewer physician tweets available, so it is possible that with more tweets over a longer period of time, the sentiments of the anonymous physician tweets would result in a comparable pattern over time, that of the larger COVID-19 data set. A majority of tweets for both data sets were assessed as being positive, with no statistically significant difference in overall sentiment though. So here's where we assess the sentiments of tweets containing the 4 topical phrases of interest. The small size of the physician tweet data set prevented us from assessing topic-specific sentiments over time, but the overall proportions are listed on the right of the slide. The proportion of tweets for both data sets trended positive with regard to PPE and telemedicine, while for both data sets, unemployment and racism, as might be expected, trended more negatively. It's interesting to note what's happening in terms of current events during these timeframes. For example, in the top right graph for unemployment, there is a spike in negative sentiments when unemployment rates reached their highest starting in April through May, and then declined in June. So through our work here, we found that the discourse of anonymous physician tweets differs from the larger, more general Twitter discourse when it comes to COVID-19 topics. Also, the ability of the physicians to maintain some level of anonymity on Twitter holds some importance. Previous studies have found that anonymous social media data is different from more general social media discourse, particularly during a social crisis. It tends to result in more honest opinions due to less risk of repercussions. It's as Oscar Wilde says, give him a mask and he will tell you the truth. Literature quotes aside, it would be beneficial for future work to improve sentiment analysis tools so they are capable of assessing anonymous physician social media data, since this type of data and these types of masked Twitter handles are not likely to disappear in the future. Along those lines, since human language is complex, sentiment analysis can be difficult to get right. We did a manual analysis of physician tweets and scored them positive, negative, or neutral, as the Vader sentiment analysis tool would do. And upon comparison with Vader, the F1 score was 0.52 with precision at 0.63 and recall at 0.56. And you can see from the examples in the table that there is perhaps room for improvement. Of course, there's room for improvement in our methods as well. First, the sample data of physician tweets was very small. Also, while physicians were given some level of anonymity and posting, the administrators of the page know who the physicians are. So they're generally unknown to the public, but not totally anonymous. And last, which I touched on a bit already, is that sentiment analysis tools are not necessarily made to assess this unique type of social media data that has more clinical language. So in future, lexicons should be created that are tailored more toward this type of data. Also, it would be a benefit to mine the public Twitter discourse for general healthcare professional data so that that could then be compared to the anonymous physician data. Overall, social media is a rich, if not messy, source of information and can help in gaining better insights to frontline healthcare workers' perspectives, which can in turn improve patient health outcomes and public health strategies, particularly during social crises like COVID-19. And just a huge thank you to these people and these groups, and I'll take questions now.
All right. So thank you very much for that talk there, Catherine. And we're now open for questions. So let's take a look here. Great. So we have a question here about in table 4, the false positives and the false negatives are pretty striking. Can we trust these sentiment tools at all?
You know, that's a great question. I think that, part of the idea of this project overall was to sort of test that exact sort of question is, will these sort of sentiment analysis tools do a great job of analyzing these specific types of tweets where, there's a lot more sort of clinical language. It's coming from, physicians who are working on the front lines and using specific vocabularies that perhaps these sentiment analysis tools aren't prepared to assess. And so part of that was, we wanted to show that there is room for improvement here, I think. And it's really interesting to see, and I think would be interesting in the future to look at other sentiment analysis tools and see how they compare to these ones that we looked at. So yeah, great question.
Great. And we do have another question coming in, which is, can the general tweets be used to get a sense of compliance to the COVID guidelines by people?
That's a great question. I think that general tweets being used to get, sense of compliance, I think that there has been a little bit of work on that. I haven't really looked into that particular topic specifically, but I think Twitter as a source for understanding, public discourse and what they are doing. I think, you can imagine somebody posting a tweet about, hey, I got a new mask today. Here it is. I'm wearing it. You know, I went to the store and saw somebody without a mask on, or, you know, things like that. I think definitely, yes, that could be done.
Great. I think we may have time for one more question. Maybe I'll ask a question if we don't get others. I was just curious, like, what when you think about the future, what other applications of this work do you see are kind of the next steps toward applying it, given all the lessons you've learned from this work?
That's a really great question. I think that, you know, I posed a couple of examples of what we'd like to do in the future, or what we'd like to see, not that we have to do it, but that other individuals might be willing to do, would be to create sort of, you know, comparing sentiment analysis tools that already exist, and then perhaps using the knowledge from that to create one that is maybe catered more toward clinical language. And particularly, you know, language on social media data, with a pandemic hitting, and the, you know, social media being a huge, you know, basically way of communication in the current day. I think it would be a great thing to do, and I think that in the future it's something that would be really useful in terms of understanding what is happening. I think, you know, I'd like to think that this will be the last pandemic we see, but, you know, that being true, that may not be true. And so I think this type of work in Twitter offers kind of, you know, as I said, it's messy, but it offers some very unique insights that we may not get from, you know, electronic health records or other sources of data out there. So yeah.
Great. Well, thank you very much. Let's go on then to our next talk, which is on protein sequence models for prediction and comparative analysis of SARS-2 human interactome. There'll be a talk by Meghana, and I'll pass it over to you.
Hi, I'm Meghana Shirsagar from the AI for Health team at Microsoft. Today I'm presenting joint work with Nurit Asnina, Jeff Law and TM Murli from Virginia Tech, Michael Ward and Greg Bowman from Folding at Home, Judith Klein from Colorado School of Mines, and my colleague Juan Lavista Ferres. This work is about protein sequence-based models for prediction of interactions between SARS-CoV-2 and human proteins. Human coronaviruses such as the SARS-CoV-2 virus, which is the causative agent of the ongoing COVID-19 pandemic, are associated with high morbidity and mortality. Understanding the molecular interactions between SARS-CoV-2 and human cells is hence key in developing therapeutics. However, for a newly emerged virus such as SARS-CoV-2, experimental approaches such as proteomics-based approaches can take several months to years and are resource intensive if you try to do small-scale experiments. Computational approaches fill this gap by using available information such as the DNA sequence of the virus, which is a lot easier to obtain, and then building models on top of these. The goal of this work is to build a predictive model for protein-protein interaction prediction between SARS-CoV-2 and human proteins. Now, why are these protein-protein interactions important? Well, because at every step of the infection process, right from when the coronavirus particle enters the human cell and to replication and so on, the virus targets specific human proteins in order to achieve its task. So if you look at the cartoon example on the right side of this slide, I have shown how this SARS-CoV-2 particle is using the spike protein on its surface to go and physically bind to the ACE2 protein on the human cell surface. ACE2 is a transmembrane protein, and as a result of this physical interaction between spike and ACE2, the virus is able to penetrate into the human cell. Now, why is it challenging to study these protein-protein interactions? Well, because the space of all possible interactions is very large. So if you had to experimentally validate around 600,000 protein-protein interactions, which is the number in the case of human SARS-CoV-2 proteins, then it's a very large space to explore. And for computational approaches, this is made difficult by the fact that there is very limited data available in general on virus-human protein-protein interactions, because this is a cross-species protein-protein interaction. And also, virus protein structures are not very well known. There aren't that many virus protein structures that have been studied. In particular, this is the case for newly emergent viruses such as SARS-CoV-2. Hence, we derive an approach that is based purely on the protein amino acid sequence. So we developed two types of models. One is a supplemental model, which uses data from the initial SARS-CoV-2 proteomic studies to predict additional protein-protein interactions. And the other is transfer learning-based models, where we use data from other viruses. The data sets that we use are mainly for SARS-CoV-2 are from these two papers. One is the Gordon et al. Paper, which has around 300 interactions. And the more recent study from Stukolov et al. Is used to validate the predictions that we obtain by training a model on the initial data set from Gordon et al. We use a publicly available database called VirusHostNet for data on other viruses. Our machine learning pipeline takes as input a pair of proteins. So a pair is an example where a pair consists of one virus protein and one human protein, and the output is whether they interact or not. So given known interactions, such as the data from Gordon et al. Or VirusHostNet as known PPI, for each of these interactions, we generate features where features for the human protein are concatenated with the features for the virus protein to get a joint large feature vector, and then we build a model on top of it. We use the generalized additive model with interactions in our work because models from prior work and in also recent work, which are deep learning based, do not allow us to interpret what the model has learned or to reason based on sequence feature combinations, which feature combinations are important for an interaction. So the generalized additive models in general assume that the output is an additive model over the individual input features. And generalized additive model with interactions incorporate an additional term which involves interactions between features xi and xj, and a function is learned over these interactions fij, which is a nonlinear function, and it is learned using a bagging algorithm. We use protein sequence derived features, monomer, dimer, and trimer features. In addition, we use physiological properties of amino acids to derive conjoint triad based features. Overall, we have around 17,000 features per example, that is per protein pair, and we prune them down to around 7,000 features by doing a feature selection step that removes some of the trimer features. In this example on the right, I'm showing the types of features that would be derived for these for a given protein pair. Now, given this protein sequence-based representation of the input, we want to see what does protein-protein interactions data look like if we visualize it. This is a two-dimensional representation of protein-protein interactions where each dot is a virus-human protein-protein interaction, and it comes from one of these 4 viruses. This allows a biologist to compare interactions data sets across viruses. So for instance, here you can make the observation that this big cluster at the center, which consists of a lot of overlapping points from SARS-CoV and SARS-CoV-2, is a cluster of interhologous interactions between these two, involving homologous proteins from SARS-CoV and SARS-CoV-2, and potentially homologous proteins which are the human binding partners. We do further analysis of this plot to find clusters, and we find that there are functionally similar human and virus proteins that appear in these clusters. An interactive visualization of this plot is also available on our GitHub page. In terms of experimental setup of our supervised machine learning pipeline, we do a tenfold cross-validation based setup, and we evaluate the prediction performance on both SARS-CoV and SARS-CoV-2. Since there is no negative interactions data available, we use heuristics that involve combining some information, some protein domain information from the negatome database, and randomly sampled pairs of virus and human proteins. The positive to negative class ratio that we use is 1:10, and we compare to a couple of prior works, random forests, and a very recent deep learning based protein sequence model called TAPE. Since this is a class imbalance setting, we evaluate and compare area under the precision recall curve. In terms of predictive performance, we find that our model does best for SARS-CoV-2 predictions. The deep learning based TAPE model is also very competitive. And another interesting observation is that the transfer learning based approach is very good at retrieving interlogs, which is shown by a high early precision value. We further analyze our predictions by comparing the set of predictions we get from on these 600,000 pairs of proteins to a recent experimental study from Stukolov et al. And we find that the positives in our predictions are significantly enriched. We also do Go term enrichment analysis, the results of which can be found in the paper. Since our model is an interpretable model, we can look clearly at the combinations of amino acid sequence based features that result in predictions, predicting interactions. We also do a brief structural analysis of our model where we consider virus proteins and we try to localize which specific region within the virus protein is responsible for contributing to the interaction. In the case of NSP16, we find that out of 5 pockets that NSP16 has, 3 of the highest scoring regions that our model finds are inside 3 of these 5 pockets. To conclude, we built a model for predicting protein-protein interactions between SARS-CoV-2 and human proteins, and we present a way to visualize this data. And we also show that our interpretable model does better than previous popular models. The predictions and source code from our work are available on our GitHub page. Thank you.
Right. Well, thank you for that talk, Meghana. That's really some interesting work that we got a couple questions on already here. So let's start with that. So how does your method compare to methods based on sequence co-evolution, evolutionary couplings?
Hi, yeah, that's a very good question, but I've not had the chance to compare to methods that use coevolution based information. Did you have any particular approach in mind?
Well, I think if the questioner has one, to please post it on the Q&A, and we'll address that. I think right now it sounds like it's more of a general question. Okay. In the meantime, let's go on to the next one. GAM and GAM transfer seem to have better performance in SARS-2, but not SARS-1. Why? Can you explain a little bit more about that?
So I have a sense that it might have to do with how many, I mean, in general, when we evaluated how the generalized additive model is doing on the test data, we found that it tends to exploit the fact that there are a lot of homologs and a lot of remote homology in the training data and the test data. And I have a sense, I mean, my feeling is that SARS-CoV-2 has the interactions data that we used in particular. I think SARS-CoV-2 has more such pairs of interlogs, that is not something as strong as an interlog, but then at least protein sequences where there is more remote homology, whereas SARS-CoV-2 had fewer such interactions. And so the GAM based models, they just tend to exploit this better. They're able to exploit this better for SARS-CoV-2, and so they do better on SARS-CoV-2.
Right. And we do have a clarification on the last question, just to reiterate that. That was, how does your method compare to methods based on sequence coevolution, evolutionary couplings? And the reference was to see the work of Debbie Marks's group.
I see.
That helps on kind of narrowing that down.
Yeah, certainly. It would be something we would consider in our future work.
Yeah, great. So let's see if we open it up for any other questions here on this section. All right. Oh, I think I saw. Nope, that was a different question. Okay. So I mean, I think one thing that was interesting, right? You applied it to different areas, but are there other ones that, other areas that you think that it could apply in the future? Like, what would be kind of some of the next steps, you know, if someone were to take an actionable item using this?
So we are working with some experimental biologists right now. So we have some collaborations with people that are developing molecular dynamic simulations. So we are working with them to see if there is a way to identify which regions of the virus protein are interesting from our model, and then for them to look at their simulations data and see if there is some biological reason for it. And based on those insights, working with biologists to see if we can prioritize some interaction studies. So basically doing small scale protein-protein interaction experiments using the predictions from our model.
Right, that's great. All right. So next up, just wanted to announce a little bit that, remind people that there's going to be the Parasitic Symbiont Awards ceremony. That'll take place after the workshop. So that's going to be 1 p.m. Pacific time, or 4 p.m. Eastern time. And so feel free to join in for that. At this time, we're going to be going into basically a discussion session, and there was a link that was just sent out for that. So for those who would like to go for a more in-depth discussion, feel free to click on that link and we'll join you there in a little bit. Just as a reminder also, there are another 3 papers that are in the proceedings on this topic for this session that were presented as posters. And so if people want to explore that in more depth, to feel free to read those proceedings that are online. All right, so looking forward to joining those who are going to the discussion section that is coming now, and hope everyone has a great day. All right, bye for now.